Hello everyone. Today let's talk about sexual reproduction in flowering plants. Flowering plants are also called angiosperms. What are flowers used for? They are a source of beauty. They are for aesthetic purposes. They are for ornamental purposes like for decoration. They are used in our social life like we give a bouquet to somebody. They are used in religious purposes like offerings in temple. They're also a part of our cultural values like um, people put garlands to each other on marriage. That's a cultural occasion where flowers are used. So, in your textbook, there are questions where you have to name a few ornamental flowers or flowers used for ornamental or aesthetic purposes. So, you can name any of these 5 flowers. And the same flowers can be used for social and cultural value also, like marigold, jasmine, rose, orchid, daisy, or sunflower. Now, what is floriculture? Floriculture is the cultivation of flowers, of course, for a monetary benefit, and for these purposes, like for ornamentation or for some social purposes or religious purposes, these flowers can be used. This is called floriculture, that is, cultivation of flowers. Now, let's go into the chapter details. Like, we have to talk about the male reproductive structure and the female reproductive structure. First, let's focus on the male reproductive structure. That's also called androecium, or it's also called stamen. A stamen has 2 parts. As you can see here, a stamen has anther and then it has filament. So these are the 2 parts of androecium or stamen. So anther is located at the terminal portion and it is mostly bilobed. That is, it has 2 lobes. It is bilobed. It has 2 lobes. It is generally bilobed. It can be multilobed also. And this is the filament. Filament is the long, slender stalk, and here it could be attached to the thalamus of the plant. Thalamus is the swollen base. So here you can see this is the thalamus. This is the androecium. That is the male reproductive part, and this is the female reproductive part. All these are attached to the thalamus. So thalamus is a modified stem. And when I have talked to you about this type of anther, anther always need not be the same type. Anthers can be of different type. Stamens can be of different type. The male reproductive part, depending upon the species, the structure might vary. The shape might vary. Shape of the anther, length of the filament, it can vary. Here I have also shown that different pollen grains from different species will look different. Now when we come back to anther, let's discuss about anther in detail. Anther, I said, it is bilobed. It has 2 lobes. And even if you cut open the transverse section of the anther, you can see that there are 2 thecas, 2 sacs in each lobe. That's why it's called dithecous. It has 2 theca in each lobe. And then there is a longitudinal groove running around lengthwise, not around, lengthwise. It separates the theca. Then, and if you look at it, it has a tetragonal structure because it has 4 sides. 1, 2, 3, 4 sides. It's a 4-sided structure. And there are 4 microsporangia located at the corners. So this is 1 microsporangia. Inside the theca, there is microsporangia. This is called microsporangia. And the tissue inside it is called sporogenous tissue. The tissue inside is called sporogenous tissue. This has many cells, and these cells will give rise to pollen grain in future. During development, these tissues will convert into pollen grains. How is this happening? Let's see in detail. When we look at the transverse section, you can see Microsporangia, and it has the sporogenous tissue or the sporogenous cells. So this Microsporangia develops into pollen sacs. So this Microsporangia, this is from the top when we look into it, but actually these are all throughout the anther. I'm sorry. It's all throughout the anther. It's a big sac filled with pollen grains. So how does the sporogenous cells convert into pollen grains? That we have to study in detail. So when we look into the microsporangia, it has 4 walls. Each microsporangia has 4 walls. The outermost wall is called epidermis, and then below that layer you have endothecium, and then you have middle layers, and the innermost layer is called tapetum. So outermost layer is called epidermis, then you have endothecium, and you have many middle layers, and then the innermost layer is called tapetum. So these are the 4 walls. The outer 3 walls helps in protection and helps in dehiscence. Dehiscence means it is the opening of split open of the anther and the release of the pollen grains. So for that, the role played by these are the 3 walls that play the major role in dehiscence and protection. Then what's the function of the tapetum, which is the innermost layer? The innermost layer or tapetum, that is very much near to the sporogenous tissue. It provides nutrition or nourishment, and so it has a dense cytoplasm filled with nutrients, and it also has more than one nucleus. So when a cell, inside a cell there's a nucleus, and when the nucleus divides without the cell dividing on its without the cell division, only the nucleus divides, it's called in such a case you will get more than one nucleus. Here if the nucleus again divides without the cell dividing, then you get many more nucleus. So this condition is possible in which the cell does not divide but the nucleus divides. So in tapetum you can see such a phenomenon. So here the tapetum has a dense cytoplasm, has more nucleus, and it provides nourishment for the sporogenous tissue. This is the sporogenous tissue. It has many cells. These are the cells which convert into pollen grains. So here you can see they are converted into pollen grains and they are released by dehiscence is the process of release. Now if you look here, the sporogenous tissue undergoes meiotic division. It undergoes meiosis. The sporogenous tissue has many cells. These cells are also called PMC, that is pollen mother cell. These cells undergo meiosis and they give microspores. This process is called microsporogenesis. That is, genesis is the formation. How is microspores formed? From PMC. Through meiosis, microspores are formed. PMC means pollen mother cells. These are the cells present in sporogenous tissue. And this process is called microsporogenesis. and these microspores develop into pollen. Okay, so one PMC cell can give rise to 4 microspores, and they are in a cluster together. So that's why it's called microspore tetrads. Tetra means 4. Later on, when they grow and develop and mature and dehydrate, they separate from each other and they form pollen. Individually, they are called microspores. Together, they are called microspore tetrads. So here you can see this is the transverse section of the anther, and here you can see 4 thecas. Let me see, look into one theca, and here you have the sporogenous tissue, and inside one cell, that's the pollen mother cell. It's in diploid stage. Diploid means the basic set of chromosomes is in double. Then it undergoes meiosis, has 2 stages, meiosis 1 and meiosis 2. By the end of meiosis, you get 4 kids or 4 cells, 4 microspores. So in a cluster, they are called microspore tetrad. And this microspore tetrad, they mature and form pollen. So it's called microsporogenesis. So they mature, dehydrate, and dissociate from each other, and they develop into pollen grains, and they are released outside. This process is called dehiscence, the process of release of pollen grains. So we have started by saying that there are 4 thecas, and theca is also called microsporangium, and the microsporangium has 4 walls, and inside the tissue is called sporogenous tissue, and inside sporogenous tissues there is pollen mother cells. Pollen mother cells undergo meiosis to give microspore tetrad. They mature, dehydrate, and dissociate from each other to give pollen. So we started from theca. Theca has 4 microsporangia. In microsporangia there are 4 walls, and inside there is sporogenous tissue. And then inside the cells are called PMC, that gives microspore tetrads by meiosis, and then they develop into pollen. When we look at the pollen grains, they may be of different shapes and sizes, but generally they are spherical and the diameter is 5 to 50 micrometers. And it has 2 walls, exine and intine. Exine is the outermost layer. It is made up of one of the most resistant organic material called sporopollenin. It can withstand high temperatures, strong acids, strong alkalis, and no enzyme has yet been discovered which can dissolve sporopollenin. So it is preserved as fossils also, and because of this exine coating, it can stay for about 30 minutes or even months or many years if it is inside fossils. I mean fossilized. So then how does the fertilization takes place? There are germ pores. These are apertures or gaps in the exine where there is sporopollen is absent. And what about intine? Intine is the inner wall, and it is thin. It's not that strong. It is just made up of cellulose and pectin. And It has cytoplasm. It has plasma membrane. And if you see, you can see here vacuoles, and there is a nucleus. This is called vegetative nucleus. And you can see another nucleus called generative nucleus. And you can see a cell inside this pollen grain again. So let's look into the details. Inside a pollen grain, there is a vegetative cell. This is called the vegetative cell. And it has a vegetative nucleus. Apart from this cell inside a cell, there is a generative cell and a generative nucleus. So there is a vegetative cell, that's the larger one, and the nucleus need not be spherical, it can be irregularly shaped, and it has abundant food reserve. And then inside this vegetative cell is floating the generative cell. The generative cell is floating, it is spindle shaped, it has a spindle shaped nucleus also, and it is small. So here this is called two-celled stage because there's a vegetative cell and its nucleus. Apart from that, there is another cell floating inside the cell. It's called generative cell with its own nucleus. This is called two-cell stage. So most of the pollen grains are shed in this form, in this two-cell stage. 60% cell plant species shed their pollen grains in this stage. But sometimes in 40% of the species, as we have discussed here, this is the pollen grain. This is a vegetative cell with vegetative nucleus, generative cell with generative nucleus. This generative cell again splits by this time by mitosis because it is just duplicating itself into two sperm cells. This is called three-cell stage because you can see the vegetative nucleus, vegetative cell, two generative cell. This is the three-cell stage. So we'll discuss about female reproductive organ in flowering plants in the next video. Thank you.